Could you please introduce yourself to us?
I'm a member of the Whitehead Institute for Biomedical Research. I'm also a professor at MIT and a member of the Howard Hughes Medical Institute. I'm going to talk to you today about some very interesting new developments in the world of protein folding.
What is the protein folding problem?
Protein folding sounds like a rather esoteric subject, but it's actually really central to biology. The basic problem is this: the linear information that codes for us, codes for everything, every organism on the planet, is DNA. And it's one long string of code. And it doesn't do anything terribly interesting. What it does is it codes for proteins. And basically, proteins are us. We're all made up of proteins. And all of the things that we do that are interesting primarily are due to either proteins directly or the action of proteins. But the difficulty is that the proteins are very complicated structures. When they're first decoded from the genetic information, the DNA, they start out as long strings of linear amino acids, polypeptide. And that does nothing. It has to fold in this very, very complicated sort of origami-like way into a very, very specific structure for it to function. And getting that done in this incredibly crowded environment of the cell is actually quite difficult. And so there are a very large number of human diseases, for example, that are due to problems in protein folding. And my work has moved from doing really very, very basic biological experiments and biochemical experiments in test tubes, aimed at trying to understand really the very basics of how proteins fold and why they misfold and what goes wrong, to now working on much more complicated systems